Hello, and thank you for joining Cure Alzheimer's Fund today for this important presentation. I am Meg Smith, CEO of our organization, and I hope you will allow me just a few minutes to introduce CureAlz. We were founded with one mission: to fund research that will accelerate prevention, treatment, and cure for this disease. To that end, since 2004, we have deployed more than $200 million toward extraordinary research pursued by brilliant and determined scientists around the world. Our dedicated donors make all of this possible, people who share our drive and commitment to end this disease and its terrible impact on patients, families, and our communities. We are grateful for their trust in us, and we are determined to honor it by finding and funding the vital research that is the only way to achieve progress. Our board of directors and trustees hold us accountable to our mission. They and certain other supporters cover all of our operational overhead. This generosity and vision mean that our other wonderful donors know that 100% of their gift goes directly to our research program. At CureAlz, our only goal is to find solutions to help the Alzheimer's community. We do not pursue profits or financial returns. All of our scientific decisions are made by active researchers who themselves are at the forefront of discovery. Our research leadership group guides our scientific decision-making. They are experts in scientific specialties relevant to Alzheimer's who lead labs at top academic research institutions around the globe. They determine what investigations we will support, always ensuring we pursue the ideas that science and data tell us are the most likely to make a real difference for current and potential Alzheimer's patients. Thank you again for joining us to hear about the science and progress that matter in Alzheimer's. We hope you will continue to join us in our mission to end this disease.
So long time ago, I started my college degree back in Australia, and I was really interested in anatomy and cell biology. The whole idea of just looking at how individual cells in the brain may interact with one another in a normal physiological sense, or how that interaction may change in the context of pathology. And this is really going to be a common theme throughout all of the little vignettes of stories I'm going to tell you today is we are very much a disease agnostic lab, but when you break down what we're doing, we're really looking at how these cells interact with one another. I then decided, while I'd done some work on peripheral organs like the kidney and the lung, that I really was taken by neuroscience and did a secondary bachelor's degree, really focusing on neuroscience. And then, sort of not happy with the ability to get drugs into the brain, I decided to do my PhD in pure pharmacology. But this whole time, I was really struck by the fact that the cell biology was really the most interesting academic part of the research for me. So in 2012, I moved over to Stanford and joined the lab of Ben Barris, and there I jumped full throttle into studying glial biology. And then in 2018, I moved to NYU, and just a few weeks ago, I was promoted to associate professor. But one of the roles that I get to do, which is really quite exciting, particularly being quite, I can't say as young as I used to be, but being quite young still, is I co-direct a clinical basic science interdisciplinary neurology center that was founded by a donation from Monika and Devin Parek. And in this role, it enables me to actually partner clinical scientists who have a really interesting clinical aspect of research, but they don't have the molecular and cell biological tools to investigate it, and really high quality molecular cell biology researchers who may not have access to those clinical patients. And so I spend a lot of my time matchmaking to make sure that the best science can possibly happen across a whole host of different diseases, including Alzheimer's disease. But I also have a bunch of other hats that I put on throughout my time. So I'm part of the research leadership group at Cure Alzheimer's Fund, which is really exciting, get to interact and help review and pick out the latest and greatest research that can be happening. But I also do a whole bunch of other things. So late last year, we set up a new research foundation for studying ALS with Tambourine Capital. I also sit on the research leadership group of Glaucoma Research Foundation, because we do a lot of work in that space as well. And then I also get to hand out money through membership on the leadership board of Global Bioaccess Fund, which is a venture capital fund. And then I'm also involved in two companies, one called Synapticure, based here in the US. I was about to say here in the US, but I'm not in the US right now, which is really based on using patient-derived stem cells to look at patient-specific therapies, largely targeting amyotrophic lateral sclerosis. And then a company that I helped found based in London called Astronautics, which is really focused on targeting astrocytes, the cells that I'm going to tell you about today, in the context of Alzheimer's disease and other dementias. So lots of things busily going on in addition to all the really exciting stuff that's happening in the lab. And that's what I want to tell you about today. So I also just want to start by, I generally like to talk to people and say that, this is a really difficult job, but, we have these interesting ideas. The goal of a scientist is not to prove that your ideas are correct. The goal as a scientist is to test if those ideas are true or not. And sometimes it's really hard to come up with what is the best question that we should be testing. And so one of the big problems that I have that we sort of discuss in my lab on a regular basis is what exactly is neurodegeneration? As a very basic premise, this is what Alzheimer's is. It is neurons dying. But what is it when we say this word, and what do we really mean? And so I jumped on Google because Google knows everything. And I just asked Google, what is neurodegeneration? And it told me it's generally a umbrella term for a whole host of different conditions in which neurodegeneration can occur that's going to affect the neurons in the brain. And largely, of course, we're talking about this in the context of humans. And when those neurons don't properly function or don't properly replace themselves, they can become damaged, they can die. And one of the problems that we have as older humans is we are not very good at making new neurons. We can learn new things and we can make new connections. We can't readily make new neurons. And so once they die, they are gone. And so what we're really interested in is whether or not we can replace what's there, or we can stop the ones that are dying from dying in the first place. So the reason I bring that up is this is a very old diagram. You may have seen several versions of this over the years listening to Curel's discussions, but this is a very basic diagram that's aggregating a whole host of studies, many of them funded by Curel's, that's looking at, along the bottom here, the clinical stage of the disease, so where we're cognitively normal, where we all are now, as we're becoming mildly cognitively impaired, and when we're having full-blown dementia later in life. And we're looking at the magnitude of a whole host of these different biomarkers that we may use to determine whether or not a patient who presents to the clinic has Alzheimer's disease. So for instance, we may have in this purple line here, we know Alzheimer's disease is associated with a loss of memory, and that memory loss gets greater as the disease progresses. And I already mentioned things like here in blue, we know we have aggregation of pathogenic proteins like tau and aggregation of other pathogenic proteins like amyloid beta. And you may have heard of a number of different clinical trials and drugs that have come to market now that have been targeting these amyloid deposits in the hopes of actually improving the response to these, or slowing down the progression of this disease. And so with that, I normally say, well, these are all lots of big words, but what is it that we actually might know about the brain? We may argue that I could know more than you, but I would argue you know a lot about the brain as well. And there's things about the brain that you know that I don't know. And so I always like to ask people what it is they know about the brain. And people normally say, well, it's in my head. If I knock my head, I tend to have headaches and it can be really problematic. And we know Muhammad Ali had too many hits to the head and had some degeneration and contracted Parkinson's disease. So then I say, okay, well, do you know about neurons in the brain? And people say, well, yes, no, the cells in the brain, and there's something about electrical activity, and they're really important. They sort of make us do all the things that we do. And then I ask people if they know about glia. And this is normally when I see that there are crickets in the room and no one says anything. And then I sort of have to ask them, well, that's fair. Should you even care about this, right? If we all know about neurons and it's neurons that are dying, should you care about anything else that's happening in the brain? And I'd like to say that, yes, you should care. We've been working on this for a long, long time. I said that we're very disease agnostic. For a number of years ago, we highlighted that there's a population of these glial cells that might be driving the death of neurons in Parkinson's disease. With some work that we did with David Holtzman, who's another Curel's funded researcher and also a member of the research leadership group, we're looking at this particular gene, apolipoprotein E, that you also may have heard of, which is highly present in these astrocytes. We worked for a number of years looking at these neurotoxic astrocytes that I'm going to tell you a little bit about today as well. And the idea that this actually might be one of the very earliest evolutionary responses to pathogenic inflammatory infections that the body gets. And it just so happens that in the context of Alzheimer's disease, this normally helpful response has become really pathogenic and problematic. So who really cares, right? I mean, this is the take-home question. I care about glia, but how can I make you care about glia? And how can I make other students care about glia? And how can I make generally people at the NIH care about glia as well? So what I'd like to do with you now is we're just going to run a very simple little experiment. We're going to make a brain together, which seems like an absurd thing to do via Zoom, but This is the weird and wonderful world we live in. So what we need in our brain is these neurons that namely everyone knows about, these electrical cells. They're everywhere in the brain. There's many, many of them. They make us live and love and feel and think and move our arms and do all those things. And without neurons, nothing happens, right? It's the most important thing in the brain. It is the output cell that makes everything happen. But as you can see on my block here, they're about 40% or so of the cells we have here. Then, of course, we have to bring those neurons nutrients. So we have to have blood vessels. And so those blood vessels are made up of what I would call endothelial cells. We can just call them blood vessel cells among friends here. And these are really important because in the brain, they're particularly different to all the cells elsewhere in the body because these endothelial cells in the brain do not let any of the molecules that are in our bloodstream exit into the brain. If blood does exit into your brain, that's called a stroke, and that's really problematic. And there's a reason that we have to have these very tight, we call it a blood-brain barrier, tight blood vessels in the brain, because we don't want any toxins or pathogens that we may ingest getting into the brain to damage those very precious neurons. There's also some other cells. These all start to have really weird and wonderful names now. So there's oligodendrocytes. These are the cells that will actually wrap around your neurons to make sure that they can function effectively. I like to think of this as the same amount of rubber or plastic casing you put around an electrical wire to make sure the electricity doesn't get out. This is to make sure that the electrical signals in the neurons don't get out. And then we have a number of smaller cells, which we call oligodendrocyte precursor cells. They can turn into oligodendrocytes. You have more of them when you're young, less of them when you're old. But there's now some evidence coming out that these OPCs may actually have immune functions as well. Then we have microglia, the resident immune cells of the brain. This is actually what protects our brain from all these pathogens. They can move around. They're highly motile. They gobble up all the baddies as they come along. But like astrocytes, as you'll learn today, they can also become problematic in the context of disease. Then we get a little bit weird. And there are a couple of groups working on this in the Curals family, which is really great. So people like Yoni Kipnis is working on these meningeal macrophages, which are specific immune cells that sit on your brain. There's also epiplexus cells, which are specific immune cells that sit in the fluid in your brain, the cerebral spinal fluid. Then there's skin cells that make up a particular tissue called the plexus or the choroplexus, and that's what makes all the fluid in your brain to make sure it doesn't smash against your skull. Then we have pericytes, which are kind of like smooth muscle cells, which is like having a muscle in your brain, and they control blood flow. Then we have all sorts of other really weird cells like tanycytes and circumventricular cell organs, and all sorts of weird and wonderful cells. But I guess this sort of take-home message from here is that we can come up with all sorts of names for things. But the bit that's missing in this that have now popped up here in green are these astrocytes. They're an extremely abundant cell type in the brain. They are absolutely everywhere. And they're really, really big in the human brain. It is estimated from some calculations people have done that these astrocytes connect between 1 and 2 million different neuronal connections we call synapses, slowly integrating those signals across the brain to make sure that the brain functions efficiently. They also deliver nutrients to the brain, and they take away waste products from the brain as well. So you may have heard in the news a lot of things about lymphatic flow or glymphatic flow. That's through these water channels that these astrocytes have to brush out all of the excess pathogenic proteins in the brain. And this is what they look like. I thought I'd give you a cartoon rather than a beautiful picture looking down a microscope. But as you may have guessed, and it's perfect that I'm in Germany for this now, astrocytes are actually named after the German term astrocyten or star-shaped cell, meaning they have this beautiful big fat cell body in the center here, and then they have all these radiating processes here in green sticking out from them. And they really are completely impregnated into the brain, and they're the one cell that we cannot remove because it makes everything else fall to pieces. Neurons we have here in blue, just like I said, these oligodendrocytes wrap around those neurons so they function properly. And here's our big brown blob of a microglia, this immune cell that can move in between all the nooks and crannies of the brain. So here's an actual astrocyte, how I look at it down a microscope. So this is actually an astrocyte that was purified by one of my trainees in the lab. It's from a rat brain. We use this as a workhorse for doing a whole host of different high throughput screens in the lab. We can screen for the effects of drugs. We can test for functions of these cells. We can also genetically manipulate these cells to say what mutations that are associated with Alzheimer's disease may be changing the functions of these cells. And we'll get to that a little bit in a few moments as well. But this is what I spend most of my day looking at. And this one I have a particular fondness for because this is one of the very first cells that we generated in my lab. But I sort of mentioned a few of those things that astrocytes do. So I thought I would actually just show you the actual data of that. This is what people have published on this. So, Astrocytes keep neurons alive. Neurons are kind of like the teenagers of the brain. They're not very good at doing stuff. They're just very loud. So astrocytes give them all the nutrients that they need. So here on the left is just an image in green. This is labeled those neurons. This is in a cell culture dish. And we can see when these neurons are grown on their own, on the left-hand side, they're very small and weak looking. But on the right-hand side, I'm sure you appreciate that they're more abundant. They're looking a little bit more extravagant. And these are grown in the presence of astrocytes. Much like teenagers that get a little bit angry and don't want to go out and hang out with their friends. Neurons are also not very good at making friends. So a lot of the connections between neurons that help these signals move across large distances are also possible thanks to a whole host of molecules that astrocytes secrete. So people like Nicola Allen, who's at the Salk Institute, who's also a member of the Curels family, has been working on this for a number of years. And again, we can see on the left-hand side here, there is a neuron grown in the absence of these astrocytes. And what we're looking for here is yellow dots that show that there's a connection with a neuron next door. And on the right-hand side, we can see there's many more of these yellow dots because there's many more of these connections being made because these astrocytes are present. We also know that astrocytes take out the trash because teenagers are also really bad at taking out the trash. Their rooms get really filthy. So the astrocytes actually come along like a good parent and they clean the trash away. So they take up all of that debris and they can actually package it up so it can be excreted back into the bloodstream where these astrocytes are wrapping around all the time. They really are a conduit from the bloodstream to the neurons and back again. They take up the food to give to the neurons. They take away the trash and put it back out into the waste disposal unit, which is the bloodstream. And then there's a whole host of other things that they do. They lay down the carpet for the neurons to grow on. They help modulation of immune function, which we're going to talk about today. They're really important in uptaking all of these neurotransmitters, these molecules that neurons secrete to passage messages from one neuron to another. And if there's too much of those neurotransmitters around, these neurons can become what we say is excitotoxic. That is, they just get too excited and they will die as well. So these astrocytes also calm these neurons down. Like a little kid who has too much sugar, the astrocyte calms them down as well. And what my lab is really interested in is not so much finding more of these basic functions that make the brain work, but we're really interested in working out what happens when they are diseased. And we call this a reactive astrocyte because these astrocytes are reacting to some sort of diseased pathogen. It could be amyloid or tau, it could be inflammation, it could be an external infection. We've also heard a lot about people worrying about SARS-CoV-2 leading to increased incidences of Alzheimer's disease or other diseases, some like multiple sclerosis and Parkinson's disease, which have a viral component as well. So I thought what I would do, normally I can say this is me sitting in Manhattan and this is a Manhattan plot, but this is me sitting in Germany and this is me reminiscing about living in Manhattan. And the reason we call this a Manhattan plot is if we look at this, it looks a little bit like the skyline of Manhattan. We have, the World Trade Center here on the right-hand side. We have a little bit of Midtown with some slow buildings. And then on the left-hand side here, we have Billionaires Row close to the park. And what this Manhattan plot is actually showing us is along the bottom here, we have each of the chromosomes of the human genome. And in the vertical axis, we have the probability of any one of these genes having a mutation across hundreds of thousands of patients that may be more likely than chance for these patients to have a given disease. And so we can see there's a number of genes in here that may be familiar. So apolipoprotein E or ApoE. ApoE4 is an incredibly high risk factor for Alzheimer's disease. And it's the largest one here. We can see it's really stacking up all of these mutations, showing that it's very common. But there's a number of other genes like cluster in here or apolipoprotein J, which is also implicated in disease. And things like IMPP5D, which is expressed by microglia, is super important in the context of AD as well. And so we take these sorts of data and we say, what cells express these genes and what is the likelihood if someone had a mutation in these genes that they may manifest with some functional deficit, these cells no longer interacting properly together, that could help us understand a little bit more about Alzheimer's disease. So I'm going to show you this exact same data in a slightly different way now. And you'll realize what scientists do a lot of times, they look at the exact same data in different ways to see if they can understand it a little bit better. So what I have here now is all of these different individual mutations, these genes showing here. And the vertical axis again on the left-hand side is showing us the probability that we may be getting this disease. And on the bottom axis here, going along the bottom, we have the probability of this, or the frequency of this, occurring in the population. So this number here of 0.5 means 50% of the population around the world has likely got these mutations. So what we can do is we can actually go into a whole host of cell type specific sequencing data, and we can say what cells express these genes and what cells may be dysfunctional in the context of this disease. In this context, it's Alzheimer's disease. So I just labeled them here for you a little bit. So the first one we can look at is here in blue. We have a number of genes that are expressed by by neurons. So things like the amyloid protein, APP, presenilin, things like this. And people can see there's a lot of genes here that are not expressed by neurons, but there are some that are expressed by neurons and a few other cells. We have a huge number of these genes expressed by astrocytes. I already talked about ApoE, and I already talked about clusterin, a gene that is mutated in more than 40 or 45% of the population. But we have genes that are upregulated in microglia and in all of those other weird and wonderful cells I told you about. Those oligodendrocytes that wrap the neurons, the blood vessels, or genes that is ubiquitously expressed across all of the cells in the central nervous system. So maybe we can take this same data and we can look at it another way now, and we can try and learn a little bit more about what's going on here. So what I have here is something that we would call a heat map. And all this shows us is, again, we have all of these genes I showed you on the previous slide listed on the right-hand side here from top to bottom. MS4A6E is a gene that's expressed by microglia. And there's a whole host here. We have APP, which is expressed by neurons again. And then what I've done, each one of these columns is showing us individual cell types in the brain, neurons, astrocytes, the weird ones like oligodendrocytes and endothelial cells. And the color just shows us how high or low this gene is expressed. So warmer colors like reds and orange are very high expression, and cooler colors like blue are very low expression. And the take-home message from this really is a huge number of these genes that are highlighted by mutations that are associated in hundreds of thousands to millions of patients around the world that have high likelihood of contracting Alzheimer's disease are not even expressed in neurons. You can see in the bottom left-hand corner here, it's only around a quarter of these genes are expressed by neurons. And so this is why I say you really should care about glia, even if you didn't know what they were at the start of today, because the vast majority of all of these mutations that are associated with diseases like AD are actually very highly or exclusively expressed in those genes, in those cells, I should say. So I'm going to give you just one little example of how we took that sort of data to try and understand what's happening in Alzheimer's disease and other diseases, and really focusing in on an interaction between two cells. And in this interaction, what we're going to focus on is inflammation is something that is all the way under us at all times. We may get influenza or herpes infection or SARS-CoV-2, or we have a headache, we drink too much, we fall out of bed, all those sorts of things that can happen that can lead to a little bump on the head that gives us some inflammation. And it's actually our immune cells that respond very fast to this. So in the brain, that's these microglia. They're really uniquely poised to respond incredibly quickly to these sorts of insults from the periphery. So they too, like astrocytes, can become reactive. But what we're really interested in is not the initiation of the response, it's the exacerbation of that response. So I told you astrocytes are around 30, 40, 50% of the cells in the brain, depending on the brain region you're in. These microglia make up around about 5% or so. The number is very small if they initiate their response, but they amplify that signal through astrocytes. And they do this by releasing a whole host of different molecules that we can systematically identify over time to work out exactly what the individual states of each one of these cells are. And then what we do in the lab is we say, well, if those microglia respond to a stimulus and make these astrocytes angry and reactive, what do they do? And in this particular story, we found that they can kill neurons, they can kill synapses, they can kill oligodendrocytes. We got very interested in this in the context of multiple sclerosis, because that's what happens in multiple sclerosis. They can actually cause the blood-brain barrier, that tight vessel barrier, to make sure none of the toxins in the periphery get into your brain. They can cause that to become what we would call leaky. They can feed back onto the microglia and have this vicious cycle of the microglia making the astrocytes angry, and then the astrocytes making the microglia angry. And they can also feed back on themselves and change the way that they may respond to any future stimuli. And this is really, really problematic because it resets the boundaries from an earlier life insult to the way we might respond later in life. And I'm sure we've all heard of examples of people we know who have been fine when they've caught influenza as a child or as an early adult, and they've been sick for a few days, and they've come back and they've been totally fine. You may have a friend or family member who's caught influenza later in life in their 70s or in their 80s, and they've presented to the clinic with some dementia episodes. And that's because this interaction between these two cell types, this inflammatory response, has altered throughout these lifestyle insults. So I also wanted to show you how we took this particular interaction, and actually I realized now looking at this, this is looking a lot like this astrocyte with all these lines popping out all over the place, connecting these astrocytes to everywhere. And this is some work that we did a few years ago with David Holtzman out of WashU. Again, like I said earlier, a member of the Curals Fund family. So what Dave had been doing, and this was actually a graduate student of his, Yang Shi, was really interested in the role of apolipoprotein E. Remember, I told you this is this super highly expressed gene. It's almost exclusively expressed by astrocytes, a little bit in microglia. And it was that incredibly tall building on that Manhattan plot. That was the One World Trade Center. And there are three forms of apolipoprotein E. We call them ApoE2, ApoE3, and ApoE4. E4 version is mostly indicative of a risk for white Western European populations, or there is some evidence that maybe it's slightly protective in glaucoma in those populations. It is less of a risk factor for people of African American or Hispanic background, South American background. But what Young has stained here for is a marker of astrocytes, this glial fibrillary acidic protein, GFAP, and the IF just stands for immunofluorescence. And what we're looking for, if there's more of this GFAP, we tend to think that these astrocytes are becoming more reactive. And in line with what we see in human patients, we can see that the mice that expressed ApoE2 had a low level of this protein. With E3, they had slightly more, and with E4, they had a greater degree of that. But we could also go even further and say, well, what about all sorts of other genes that are not just GFAP? Would they express genes that are indicative of this neurotoxic phenotype? And we saw that indeed those mice that were expressing the ApoE4 version when they got to 9 months of age, again, this is a heat map. The red color is a higher expression, express many, many more of these genes associated with this subtype. But earlier in life, just 6 months earlier, they were not expressing that. And more remarkably, if we actually look at the brains of these mice, so this is on the left-hand side, this is a wild-type mouse. We can see that this is just the cortex, we call this the outer surface of the brain. This is the hippocampus where a lot of our memories are formed and stored. And we can see that in this mouse on the right-hand side, which is expressing the apolipoprotein E4 version, the risk version of this gene, there is now this massive hole in the brain. And this is due to degeneration of those neurons. And we can see the hippocampus here has got incredibly small, again, causing it a lot more difficult to form and maintain these memories. So this is actually not just a mouse thing. This actually happens in patients as well, in human beings. So this is actually just some staining I'm going to show you now for a very specific marker of these astrocytes that can kill those neurons. And it's this marker here in red, which we call complement component C3. So it's these little tiny red dots that seem incredibly innocuous, but actually if we see those in these bright green astrocytes here, we presume that they have the ability to kill. And so here, this is just a tiny image of a single astrocyte from a postmortem tissue from a patient who had Alzheimer's disease. And down the bottom here, we've just counted up the number of the cells that were either normal astrocytes or these C3 red killer astrocytes. And we can see in the AD condition, there was many, many more than there was in the non-symptomatic or non-AD control patients. And this was true for a whole host of different diseases we looked at. It was true in Huntington's disease, motor neuron disease, Parkinson's disease, and multiple sclerosis. But the really important thing was that these astrocytes weren't everywhere in the brain. They were only in regions where the neurons were dying. The really scary bit, though, is if we actually go and just look in an otherwise healthy, aging brain, we also see them there. And we now know that this is not a problem. These astrocytes don't rampantly run around killing all the neurons in the brain. What they only do is they only have the ability to kill neurons that are damaged or otherwise susceptible. So now we have two targets here where we can try and keep those neurons alive by making them less susceptible, or we can keep them alive by stopping the astrocytes from killing them. So that gives us two shots on goal, which is really exciting, just by investigating this single gene mutation that was really interesting in the context of AD, that was highly expressed in a cell, and we saw this interaction between the immune system and these astrocytes. So this is another example of exactly what I look at when I think of neurodegeneration. This is actually us looking down a microscope now. So I'm going to talk you through this data as well, because I think it's really, really interesting. So what we have here on the top on the left-hand side, this is actually a neuron growing happily on a dish under a microscope so we can look at it. And what we're going to do with this experiment is we're going to try and kill this neuron. And the way that we're killing this neuron is we are growing it in the presence of these astrocytes that are neurotoxic. And we can see very, very quickly, this timestamp here is 4 hours. This neuron already looks a little bit sick. And by 8 hours, this neuron has actually died. And we know it's died because we've added this cell death label here in red. It's called a Thyddium homodimer. All we have to remember is that it shows up when neurons are dying. And we can actually look across thousands of neurons in multiple different populations. We can say how quickly do they die? And actually, it turns out that they die very quickly if they come from the eye. These are retinal ganglion cell neurons that help us see. Hippocampal neurons, again, the part of the brain that's important for memory. And also spinal motor neurons, these are neurons that travel all the way down your spinal cord and make it easier for you to move your arms and legs. And so we know that all of these neurons are able to be killed by this toxin. And so this is what it actually looks like. And this is how I watch death in real time, which is a morbid fascination that I have. But on the left-hand side, we have neurons growing happily in a dish, and these are grown in the presence of normal, happy, non-reactive, non-toxic astrocytes. And you can see these neurons are stable and they're happy. On the right-hand side, we can see those same neurons over only a course of 24 hours grown in the presence of these neurotoxic astrocytes. And here we can see that those neurons very quickly ball up and wither and die. So the model that we sort of set up a few years ago is the idea that these neurons can become damaged. Maybe they have a mutation themselves associated with Alzheimer's disease, like APP, like presenilin. It was on that big Manhattan plot as well. And they would make themselves susceptible to be targeted by this inflammatory cascade that sets up. And maybe that's what activates these microglia. These microglia can activate these neurotoxic reactive astrocytes, or NRAs. And then these astrocytes can release this toxin that will actually drive the death of these neurons. And normally this would be fine, because if you remove a single neuron, you can rewire around that hole that you have made and continue on as normal. And this happens all the time in our brains. And actually, we've shown that this is really important, because if you don't remove those neurons, in some instances, it can be really problematic. And an example we have of that is if you have a prion infection like mad cow's disease in the brain, that prion infection makes your neurons susceptible to be killed. And if you stop these astrocytes from doing that, the infection goes further in the brain. It just so happens that in the context of Alzheimer's disease, this pathway is activated in an aberrant fashion, and more neurons die than we need to have dying. So this is back to our little graph here that I had for us. These are lots of things. There's amazing people far smarter than I working on all of these particular aspects of Alzheimer's disease and doing a wonderful job, and we're learning lots from them. But I always like to think, well, what happens if we just sort of looked at the glial immune dysfunction, which happens really, really early in disease? We see these changes in these astrocytes, these changes in these immune cells, these changes in the inflammatory cells happening very much earlier in disease. And what happens if we could just open that window of opportunity we have to treat these patients? So here we know when patients present to the clinic that they have some mild cognitive impairment, we know that something is wrong. But what if we had a better way to detect even the slowest and smallest amount of amyloid that's depositing here we can detect by imaging? What if we track that back to where the glial dysfunction is? We have a much wider opportunity of window for action here. Or what happens if we looked at when we know that we have memory deficits like in dementia? We track this all the way back to where we had the first dysfunction occurring in these immune cells and these astrocytes. This opportunity window for treatment gets much, much broader, which is really, really exciting. So I got excited about a lot of stuff here. I hope I got you a little excited about a lot of stuff as well. These astrocytes, like I said, these funny cells in the brain that look like stars, are essentially the mothers of the brain. They feed neurons, they take away trash, they do all those things, which is really exciting. But sometimes in the context of disease, they can become a little negligent. And this definitely happens in Alzheimer's disease as well. We didn't talk about that today, but it's something that we do, where they lose these normal functions, which can lead to astrocytes dying. But I also told you that they can gain these functions as well, and that can drive the ability to actually kill these neurons, which is what we spoke most about today. And our goal is to be able to actually take these astrocytes and push them back into this protective state so they can actually keep those stressed and damaged neurons alive even longer, so we can slow the progression and maybe even, if we wish, stop the progression of this disease as well, which would be really exciting. So I will stop here, because otherwise I can keep talking about this for hours. But I just wanted to show you that this is the sort of exciting work that Curals has funded. And all I told you about today was these neurotoxic reactive astrocytes, one type of astrocytes of the hundreds of millions that we have in our brain. We now have identified another immune responsive astrocyte that sits around the ventricles, these holes in the center of the brain, and around the surface of the brain that respond to immune responses very quickly. We're now targeting them very robustly in these contexts of these Alzheimer's mice. We also discovered this new astrocyte that only lives on the outer surface of the brain and actually itself acts as an immune cell, so it doesn't require those microglia. We've also discovered a population of cells that is very highly proliferative and proliferates around the amyloid plaque, so it actually stops it from expanding, which is really exciting. And we're spending most of our days now just hunting down these new subtypes of these astrocytes that we see in these human patients to try and model them so we know what they're doing in the context of this disease, but also trying to work out if there are available drugs on the market already or drug targets that we can develop to try and push them back to that normal physiological function or veer them down one of these sort of more protective pathways.